Macromolecules. This time we're going to be looking at lipids. So lipids technically may not classify as a macromolecule, and then we're going to look at some different types, but we'll put them in there with the same group of macromolecules. And they, like many biological compounds, are made up of carbon, hydrogen, and oxygen. And again, you can start seeing some of the variety of things that we can build with just these three atoms, let alone throw in three more. These have fewer hydroxyl groups and fewer carboxyl groups than what you'd see in some of the other macromolecules, like the carbohydrates. And so because you have fewer of these, there's going to be less polarity to them. And if they're less polar, they interact with other things that are, they interact less than polar molecules would with things like water. So lipids, being less polar, do not interact as well with water as, say, something like a carbohydrate would. And another way of saying that is that they are hydrophobic, meaning they're afraid of water. And it is not that they're repelled water. Remember, molecules are all attracted to each other, right? Those London forces attract them all together, but it's just that water will exclude these ones because water would rather hang out with other water molecules than with something that is not polar. They are very energy rich, and they're therefore used for long-term energy storage. So different ways to build molecules that can contain energy stored in their chemical bonds. Carbohydrates are one, lipids are another. Lipids are more of a long-term energy storage, and they contain more energy in their bulk, at least per mass anyways. Four basic types that we're going to be looking at. So they're not just used for energy storage, but that is definitely one of the important roles in there. So triglycerides, also known as fats. We'll see fatty acids within there. We'll look at phospholipids and then quickly at steroids and waxes as well. So triglycerides, as the name would suggest, there's definitely going to be C3 of something in here. And they contain lots of these carbon-hydrogen bonds that again are a way of storing energy within these chemical bonds here. Really good for long-term storage, and per mass they have about twice the energy storage that glycogen would have. So fun fact would be that if you wanted to store, on average, the amount of energy you have stored in fat, if you instead wanted to convert that and store it as a carbohydrate, like in glycogen, you'd probably have to weigh up to 100 pounds more just to have that same amount of energy. So this is very energy dense. So if you want to store energy, fat is a great way to do it. They also have, these triglycerides have some role in structural support as well. So like the pads of your finger or the bottoms of your feet, they are cushioned so you can actually pick up things and have better grip on surfaces because of the layers of fat within those tissues. So the structure of the actual molecule itself has a glycerol molecules. We'll look at what that looks like and three fatty acid molecules. They're all bonded together. So let's look at these components individually and then we'll put them together. So a glycerol molecule has 3 carbons in it, and each one of those carbons has a hydroxyl group on it, an OH group on it, and then the rest is hydrogens. So you end up with these 3, let's get rid of those so you can actually see this, 3 hydroxyl groups to it. That is a glycerol molecule. 3 carbons, 3 hydroxyl groups. A fatty acid molecule is really just a chain of carbons, a whole bunch of carbons all in a row, and then at the end of it is the acid part. And remember from our functional groups, if you have a carboxylic acid functional group, that's what makes it's an acid. And so that fatty acid has long chain of carbons, that's the fatty part, and then the acid part is this carboxylic acid group at the end. Now, that's just glycerol and fatty acids. If you want to actually get a triglyceride, you've got to get that fatty acid to react with the glycerol. And they can do that with, we've seen this before, the OH functional groups. If you combine 2 OH functional groups, they can come together, and if you take an H away and an OH away, you got water, and then the remainder will be stuck together. So we call that dehydration synthesis. And since there are 3 OHs, you can combine 3 fatty acids to a glycerol to get a triglyceride. Notice there are different types of fatty acids. So this one here in green is saturated. It's all single bonds. It's holding on to as many hydrogens. So when I say saturated fats, it means that they're holding on to as many hydrogens as they can. if we look at this unsaturated one, you'll notice there's these double bonds in here. So these carbons, each carbon that is taking part in a double bond, it's not holding onto a hydrogen. Instead, it's double bonding onto the carbon beside it. So it doesn't have as many hydrogens as it could have. So it is unsaturated with hydrogens. And because of this, you end up with a sort of kinking that's happening. Remember biology, all the molecules, it's all about shape, right? And so this shape here, and then the forces with the functional groups as well, this shape here is going to make this unsaturated fatty acid act differently than a saturated fatty acid. So the reaction itself with the glycerol and the fatty acids is a dehydration synthesis, and similar to what we saw before, you're going to be taking an OH and an H, and you take them off, and you would get H2O. You do that 3 times, you'll get 3 waters, and then each of these fatty acids is now going to be connected. What used to be the acid group is now going to be connected to what used to be the hydroxyl group on the glycerol, and is now all one thing. And so that happens with the 3 of them, and you end up with this fatty acid molecule. This linkage here is called, oh, that's a better arrow, is called an ester linkage. And so that double bonded O and that carbon that's involved there, we're going to call that an ester linkage. Fatty acids themselves in a little bit more detail. So again, they're a chain of carbons. They've got this acidic functional group on the end and this chain of carbons. The 2 forms of them, first one we'll look at is a saturated one. And just quickly in here, so I say a long chain of carbons. You say, well, how long? There it is. So somewhere maybe in the teens up to 20s in terms of how many carbons are in the row there, and then this acid functional group on the end. If they're saturated, like we said, no double bonds, as many hydrogens as they can be holding on. It doesn't have any double bonds, makes the chain quite straight. And what's interesting is that will make them, and again, because the chains can be different lengths, it's not always the case, but it tends to be solid at room temperature. And these are the types of fats that you see in animals like fuzzy seals and fuzzy penguins and other warm-blooded creatures will tend to have these saturated fats as part of their structures. And some of the reasoning behind that is that, again, the body temperature of a seal and a penguin, they are warm-blooded, so they're relatively warm. So if the fats are solid at room temperature, you warm them up, they would be a little bit, they would melt, and they'd be a bit more fluid. And so if you have a warm body temperature, having a fat that is the right fluidity is really, really important. So it turns out that a saturated fat works well for warm-blooded animals. Unsaturated fats, here's some diagrams of them. Again, they have these double bonds. It doesn't have to be full of double bonds, but it's going to have some double bonding in there, and it is going to then kink the fatty acid. This makes them bulky, and therefore the outcome of that is they end up being a liquid at room temperature. So if you have a really cold body temperature, like say a fish, or maybe even a plant, and you still need your fats to be somewhat liquidy, you'd want to have unsaturated fatty acids in order to have that happen. Because if you had saturated ones, they would congeal and they'd be solid at cold temperatures. And so if you want to be able to move those cells around or do things with have more fluidity with these fats, that wouldn't happen unless they're unsaturated. So different organisms will use different types of fats in order to meet their needs for their particular conditions. You also have heard, I'm sure, of probably this one here, trans fats. And so there are actually 2 types of unsaturated fats. There's trans fats and there's cis fats. And you probably think, oh, trans fats are bad. And that is what we know about them, is that their particular shapes, the way in which they're bent, they sort of end up being sort of like a zigzag kinked to it. And so they can have negative effects on the body. They actually work with leaving cholesterol behind in arteries, and then that can block up the arteries. And so because of the shape that trans fats happen to have, they end up being very unhealthy. And they've actually been banned in Canada relatively recently in terms of potentially putting them into food. Cis fats, though, don't have that problem. They're of the right shape in that they don't actually build up in arteries. They don't cause cholesterol to build up in arteries, and so they end up being quite good. Phospholipids is another type of, as it says, lipid, and you can imagine what this lipid has going for it in the rest of the name. So a phospholipid has this phosphate group to it. There it is there. And it's very similar to a triglyceride in that we have this glycerol molecule, and it's got this fatty acid hanging off 1 and 2, but one of the carbons, so I should probably say carbon 2 and 3, but carbon number 1 here, on one end of it, we have not a third fatty acid. Instead, we have this phosphate group. And what this does is it makes this end here polar. So you end up with this fat, I should say this lipid, that has a polar end to it. This is all carbon hydrogen down here with this fatty acid here. So this is non-polar. So you end up with an interesting molecule that has a polar end to it and then has a non-polar end to it. Really, really important for life. Life consists of cells, and this is the building block of the cell membrane that contains everything that's going on inside the cell. This fact that you can have polar and non-polar held together in the same molecule allows some really complex interactions between aqueous environments and non-aqueous environments. It allows compartmentalization, and therefore you can have living cells. So this dual character, this phospholipid, the fact that you have, and remember, here's another way of saying it, hydrophobic. So this is saying non-polar, and hydrophilic, that would be polar, likes polar things, likes water. Another name for something that is both polar and non-polar, is able to interact with water and not interact with water, depending on which part of the molecule you're talking about, is amphiphatic, is another way of saying it. It's essential to the function in the cell membrane. In water phospholipids, they form this lipid bilayer, and that's when we start looking at cells. You remember what you know about cells. The cell membrane is made up of this lipid bilayer, and the bilayer is only made possible because you have a hydrophobic and a hydrophilic component to the molecules that make up the cell membrane. Here's a nice picture of it. Again, there's water outside the cell, there's water inside the cell. So how do you separate one bit of water from another bit of water so that you can have life? The answer is with a lipid bilayer. And you make a lipid bilayer by taking a molecule that has a polar end to it that likes water and a non-polar end to it that does not like water. And you put a whole bunch of those, each one of these, let's circle it a little more clearly here. Each one of these guys here is a single phospholipid. Put a bunch of them together, they put all their tails together to try to get away from the water, and you end up forming a layer. You end up forming a little compartment with an inside here, and an outside, and that's a cell. All right, quickly we're going to mention the other two fats, not too much detail, but we will see some of these mentioned later. Steroids are essentially a messaging molecule, and they are a type of lipid. Lots of carbon hydrogen components to them. They tend to have these ring structures to them, as well as some different functional groups depending on which molecule you're talking about. So here's a cholesterol molecule, as we mentioned earlier, and here's a testosterone molecule. So that testosterone hormone, you can see some of the similarities between its structure. You've got these same sort of ring structures here. They are both lipids. Some of them are used as signaling molecules, and then we have cholesterol doing its function as well. Cell membrane function in terms of playing around with the fluidity of those cell membranes and the such. We'll talk a little bit when we do cell transport, and then hormones we'll get into a lot of detail on when we look at chemical messaging. So both of those are a type of lipid. Waxes, last type, and we're really just going to be looking at it in terms of a structural component. Again, long chains of fatty acids, and then they have these rings to them. Because of this, they are hydrophobic. So if you look at the surfaces of leaves, the water doesn't stick to them particularly well. It often rolls off or beads up on them because they have a waxy coating to them. This protects the plant. The plants are constantly trying to maintain turgor pressure, so they're trying to maintain be full of water, and if they were just to lose water to the air, that wouldn't be possible. So they have this waxy coating often to protect them. That wax is another type of lipid.